this night at her end, and she needs no introduction. She is a professor of dermatology, microbiology, and immunology. She's a member of Cancer Immunotherapy Initiative. She's a co-director of SBDRC Test IT Core at Cancer Center IC Core Facilities and CSO Templian Therapeutics. And she works at Northwestern University, Chicago. So many thanks for accepting this kind invitation and for being live, ma'am. And over to you, ma'am.
All right. Thank you very much.
Thank you.
Thank you, Dr. Singh. That's a very nice introduction. I do work for Northwestern University here in Chicago. It's actually been a hot couple of days here. So I would like to talk to you about, it's very exciting to present to you for World Vitiligo Day. That's, of course, a real nice achievement that we now have this international attention to vitiligo. And so I'm proud to share this work with you that we've been doing over the past couple of years. And I'd like to especially pay attention today to the vitiligo pathogenesis and immunotherapeutics, with a particular attention to antigen-specific immunosuppression for vitiligo. Vitiligo as a disease needs no introduction to you, I'm sure. Just a few words here that you see here, this characteristic symmetrical distribution of the lesions that stands out, as well as a Kubner phenomenon on one of the shoulders of this particular patient. This is an image that I received from Dr. Westerhoff many years ago, but I think it's very characteristic. And obviously, we would love to do something about the progressive depigmentation that occurs in the skin of patients. And one of the things that we now know is that vitiligo is a T-cell mediated autoimmune disease of the skin. And that's really helped not only us, but many other groups to think about therapeutics that could restore pigmentation based on the fact that we could interfere with these immune responses that take out melanocytes from the skin. So I just want to mention, as Dr. Singh already brought forward, that I am a CSO for Templian Therapeutics, which is a startup company where we're trying to promote modified HSP70 as a treatment for vitiligo. That's not what I'll be discussing today, but just so you know where I'm coming from. So one of our earlier articles was on the etiology of vitiligo, where we already proposed that regulatory T cells might be playing an important role, or at least the lack thereof, I should say, in vitiligo. So if I can briefly go through what we think is a general overview of vitiligo etiology, or the pathomechanism. It starts really with some form of injury to the skin or stress to the skin, which can affect especially melanocytes that are essentially set up to protect us from this type of injuries, especially if it's like excessive sun exposure and things like that, which can lead to expression of stress proteins to protect the cells from undergoing apoptosis. And then they will, some of that heat shock protein can be secreted into the extracellular environment and activate the antigen presenting cells that are in the skin. So we're thinking macrophages, dendritic cells. And these can then take up the stress proteins that act as chaperones for antigens, and take those to draining lymph nodes, where they can be presented to CD4 and CD8 T cells that are reactive with the very antigens that were brought in by these chaperones. And then this can lead to progressive depigmentation when these CD4 and CD8 T cells come back into the skin. Now, in normal conditions, we all have some of these T cells that can react with melanocytes, but in normal conditions, a response like that will be stopped by the regulatory T cells that are there to end excessive immune responses when they're no longer needed. We found, however, that that's not necessarily the case, or is not very effectively happening in vitiligo. So that's really what we want to go after. What we found is that in vitiligo skin, you see very few regulatory T cells. if we compare this, for example, to control skin, now we're looking here at double staining for CD3, that's in blue, for all T cells, and then FOXP3 as a marker for regulatory T cells in red. And you see that, you know, there's about maybe 40% of regulatory T cells among T cells in the skin. In controls, lentigo maligna and malignant melanoma. But if we compare that to non-lesional, perilesional, or lesional vitiligo skin, in any of those cases, it'll be hard-pressed to find any regulatory T cells, or at least not very many. So that really means that if T cells come into the skin of a vitiligo patient, you know, they can just do whatever they want without being stopped from doing that. And so that's where we get our progressive depigmentation from, in part. So we did also find in our studies that it did not, you know, we did not find similar changes in the abundance of regulatory T cells in the circulation. I know that other groups have reported that they found fewer regulatory T cells in the circulation as well. That may have something to do, for example, with, you know, the co-occurrence of other autoimmune responses in some patients, or very frequently in patients. But we also did not find any differences in functionality of the regulatory T cells that we could extract from the circulation. So in our case, we really came to the conclusion that the regulatory T cells just don't make it to the skin. So one thing you might think about is that maybe these regulatory T cells do not have the same homing marker expression that would bring them to the skin as you would in control individuals. But when we looked at some of these characteristic homing markers on regulatory T cells, such as CCR4, CCR5, CCR8, or CLA, cutaneous lymphocyte antigen, none of them were really downregulated in vitiligo patients to any serious extent. So that doesn't seem to really explain why we don't have so many regulatory T cells in vitiligo skin. But when we looked at the skin itself and looked for the chemokines that would attract them there, the one thing that stood out is that there's a very much reduced expression of CCL22, which is like the ligand for CCR4. And we did not see much of that in vitiligo skin. As you can see, like the cells that secrete or generate the CCL22 are very much reduced in the vitiligo skin. So that could be a reason for the regulatory T cells to be so sparse. And what we'd like to do then is think of ways in which we can bring these regulatory T cells back. So to test this, we use a model of vitiligo, which is now available. We have a few models that are based on the fact that we can introduce a T cell receptor into these mice as a transgene that would be expressed on T cells so that they would all be directed against melanocytes. And in this case, we make use of a tyrosinase reactive T cell receptor that recognizes tyrosinase antigen presented in the context of MHC class 1, HLA-A2 actually in this case. So both of those, HLA-A2 and tyrosinase, have been associated with vitiligo development in genetic studies. So that makes this relevant mouse model. What's good for the use, and we'll come back to that later on, is that this T cell receptor happens to function independent of the presence of CD8 or CD4 on the T cells, so it can also function on regulatory T cells when it comes to that. These mice develop progressive depigmentation, as we also see in humans. Of course, in this case, it is in the hair follicles because that's where we find melanocytes in the mouse. But you see here that the depigmentation occurs in a relatively regulated fashion, and about 22 weeks, you have half maximum depigmentation occurring in the mice. That is associated with the loss of melanocytes from the skin, here shown in blue in the wild type mice, whereas the H3TA2 mouse does not have them anymore. And we see a little bit increased T cell infiltration as well to the skin. And we've also tested their reactivity in vitro, where we reacted them with melanocytes, either matching or non-matching, and you see that they produce interferon gamma. So we can also intermix human and mouse in this particular case. What's also important, of course, if we want to replenish regulatory T cells, is to show that regulatory T cells are sparse in the skin of this model. So we extracted regulatory T cells. I see that I have a mistake in here because this says that the top mouse is also A2 positive, H3T-A2, but it's not. So please, that number of mouse is actually not an H3T-A2 mouse, but a control. So you can see here that or that's H3T2 minus, that's what it is. So it does have HLA-A2, but it does not have the T cell receptor. So they won't depigment, and you see that in that case, you do still have the regulatory T cells in the skin, but not in the vitiligo mouse. We also know that in this model, interferon gamma is involved in depigmentation, something that's also been reported by others, such as John Harris, showing that interferon gamma is very important for these responses. And we can show this here by expressing the transgenes on an interferon gamma knockout background. So you can see the mice on the left cannot express interferon gamma, and they do not depigment in comparison to other backgrounds, such as lack of TNF alpha or perforin, where we do see depigmentation occurring. So this depigmentation is dependent on interferon gamma. And what does that interferon gamma then do? It just seemed to influence the number of T-Regs, because in the absence of interferon gamma, as you can see here, there are more T-Regs to be detected in these mice, as we show here by CD25, FOXP3 double expression by T cells in the mice. So what can we then do about this? Maybe we can say that T-Regs control vitiligo development in interferon gamma knockout mice. That's something we wanted to study in this particular experiment. So what we did here then is to look at that particular factor. So we used the interferon gamma knockout vitiligo mice, and we treat them with anti-CD25 to deplete the T-Regs. And you see that that's enough to bring back depigmentation, even though there's no interferon gamma, which essentially tells us that the role of interferon gamma producing T cells in vitiligo is somewhat redundant, because it's really about the T-Regs that we see this vitiligo occurring or not. So again, this leads us to believe that if we can replenish T-Regs, we might be able to stop the vitiligo. So here, what we're trying to do is rapamycin as an inducer of T-Regs. We do this in vitro to enhance T-Reg development in culture, so that we can increase the percentage of T-Regs, and that's what we want to do, so that we can proliferate them or expand them in vitro and give them back to mice and ultimately also to humans. We also wanted to do this in vivo, so that's what we're doing here. We treated mice with rapamycin over a period of 14 days. Then we evaluated the cells, and you can see here that that does increase the relative amount of T-Regs in the mice. And we then followed up after 56 days. So that's quite a period after the treatment with rapamycin. And you can see here that there's a significant difference in the amount of depigmentation that occurs in these mice, and that they also maintain a higher amount of regulatory T cells, even though the treatment is far behind them. So that's one approach to bringing the regulatory T cells back, and it has quite an influence on depigmentation. Another way in which you could do this is directly performing adoptive transfer of regulatory T cells. Again, in that case, of course, you would have to propagate them in vitro before giving them back if it comes to humans. But right now, we can just isolate the T-Regs, effectively activate them with a little bit of IL-2, or keep them happy with some IL-2 when they go into the mice. And again, if you then introduce these back into the mouse, first of all, you can see that the T-Regs are well maintained in those mice, and that's what I'm showing here, that leads to a decreased effector to T regulatory T cell ratio. And if you then look what that does to vitiligo development, again, this would tell you that adoptive transfer, in this case of regulatory T cells, does interfere with vitiligo development. And the number of regulatory T cells is not only reduced in the circulation, but also we see a reduced number, or an enhanced number of regulatory T cells after adoptive transfer in the skin with the stains that you see here on the right-hand side. Well, so that's regulatory T cells per se, but you can always keep in mind that if you're bringing in regulatory T cells in larger numbers into mice or eventually into patients, that you have to deal with the fact that these regulatory T cells are not specific for any particular condition. So if the patient would have an infection or something like that, they might not respond adequately, and so you'd want to actually have not only a more directed response that's only active when it's needed, but also have it be of higher affinity, because you're actually getting the T-regs to respond where they are needed and not anywhere else. What we wanted to do then is to introduce antigen specificity to these regulatory T cells and then test if this actually gives a good response as far as inhibiting depigmentation is concerned. So what that meant is that we wanted to adoptively transfer transgenic T-regs. And what we started to do is really to follow up on earlier findings by the group of Bosco et al. That used TCR transgenic T-regs generated with the exact same T cell receptor that's also built into our H3TA2 mice. At that time, those vitiligo-prone mice were not yet available. So what they did is really make TCR transgenic T-regs, introduce them into immunodeficient mice together with melanoma tumors. And then what their reasoning was that if these T-regs actually do what they're supposed to and respond to the antigen that they're directed against, that this would make the tumors grow faster, because these T-regs would be prone to home to the tumor and help the tumor cells expand. And indeed, that's what they found. But of course, that doesn't mean that it could work for vitiligo. So we wanted to follow up and do essentially the same thing, but then in our vitiligo model. And what we did then is to cross them to a reporter mouse that shows us where the T-regs are going, and that we actually have T-regs in our hands. So we now have our transgenic, TCR transgenic regulatory T cells that express EGFP in hands, and we can introduce those into our vitiligo-prone mice and then follow what happens to their depigmentation. And we did see some, quite significant effects on the dorsal side, but we weren't really happy with the amount of time that this really worked. We gave them 2 adoptive transfers here, as you can see in the beginning, and eventually that worked for a while, maybe for a few weeks, as you can see here. But then eventually the depigmentation started occurring again. But that difference also wasn't so significant. It was only found on the dorsal side and not on the ventral side. Of course, you want it to help all over. So we were thinking maybe we need something just a little bit more aggressive, and that can be found in chimeric antigen receptors. So that's something that we wanted to continue with later. But I first want to show you that the regulatory T cells that we did introduce not only interfered with depigmentation, but that also comes along with the maintenance of melanocytes in the skin. So we showed that by both TRP1 staining, by TRP2 staining, and by GD3 staining. Now, ganglioside D3 is actually an antigen that's expressed on the cell surface, and that makes it interesting for us when we want to address melanocyte viability by chimeric antigen receptors. So a chimeric antigen receptor is something that has to recognize a surface antigen. So to show the relevance for the human situation, I'm showing you here it's an older publication from us, where we showed that in the perilesional areas, you see in this case oocytylated GD3, because that's a better antibody to use for the human skin. But you'll appreciate that there is a high expression of this oocytylated GD3 in perilesional areas of the skin. We also had to show this with the regular GD3, which we did, but I'm not showing that here. And you see that this GD3 is also expressed in mouse skin, and I'm showing you on the right-hand side of this slide that there's GD3 expression in melanocyte keratinocyte co-cultures, which is really only found in melanocytes in that case. Only under very particular stressful conditions will you get expression by keratinocytes. But that means that in the areas where we really want to interfere with depigmentation, we see more GD3 expression, and that's where we want the T-regs to go. So that's what we're after in this setting. And these CAR T-regs we then generate with a chimeric antigen receptor that we have from an ongoing collaboration with Dr. Richard Youngans, who has made this chimeric antigen receptor construct reactive with GD3. What that means is that you have an antibody, you make use of the high affinity of an antibody recognition site and make a fusion protein that can also come across the members. It has a transmembrane region built into it from CD8, and then it has a TCR signaling region, and that's intracellular. And all of that is combined into a fusion product that you can then express in your regulatory T cells. And these then can interfere with cytotoxic T cell responses when combined with targets and effector T cells. So what we first need to do is then see that we can actually introduce this CAR and that it doesn't interfere with regulatory T cell function. So we have to expand our regulatory T cells in vitro, which is what we're showing here, and we can get a good amount of T-regs to work with. So we first isolate naive T cells, we then polarize them towards T-regs in the presence of TGF-beta and activation beads, as well as a little bit of IL-2 to keep them happy. And then after a while, you can actually harvest the fluorescent T-regs in this case, because they're, again, from these reporter mice in our particular case. And after retroviral transduction, we can then run them by flow to see that we actually get these GD3 CAR T-regs. So this is summarized here as well. And what you can see here is our gating strategy. So we see the time gate, you see the side and forward scatter. We select, so to select against debris, and then we make sure that we look at only single cells, and we look at the population that's alive and not the dead cells, because they tend to stick to antibody. And then on the right-hand side, you'll see the expression of the chimeric antigen receptor on our CD4 T cells. We're also comparing this by to cells that are untransduced, where you see that there's really no CAR expression, so that's a very clean difference. But you have to keep in mind that not all of your cells in that culture will remain T-regs. So a lot, or some of them are actually non-T-regs. However, you need only a very small proportion of your T cells to be T-regs for an actual suppressor response. So we're not that worried, and we put these into mice eventually. First, we want to show that this in this setting, these CAR T-regs can be antigen specific and suppress responses. So that's what we're showing here. We combined them with melanocytes and then also brought in our H3T-A2 T cells, which can only respond to A2 positive melanocytes. So we see the effectors respond only in that combination. And you will see that several cytokines that are relevant for this response are generated in higher amounts in the absence of CAR T-regs, whereas if we add the CAR T-regs, this expression of cytokines is suppressed by the effector T cells, or that are expressed by the effector T cells are suppressed by the CAR T-regs, I should say. More importantly, these CAR T-regs in vitro can interfere in an antigen-specific manner with melanocyte viability. So again, this is from that combination of melanocytes that are matched or unmatched, and then combined with effector T cells as well as CAR T cells. And you can see that in the combination where CAR T-regs are used, that there is significantly less killing of these melanocytes in culture. And that's what we really were excited about, that these CAR T-regs can even in vitro function better because of their reactivity to melanocytes, because of the expression of the CAR. So here we've introduced these CAR T-Regs into our vitiligo-prone mice and followed up on depigmentation over time. And now we can see that both on the ventral and the dorsal side, we see significant inhibition of depigmentation over time. I should mention that we learned from our first set of experiments that the CAR T-Regs, as well as that the CAR T-Regs do need to be replenished in this case. So there's of course always some improvements to be made, but I'm excited about the fact that you can always choose to reintroduce these CAR T-Regs and store them for later use. Here you can see what that does on an individual basis. So what happened to the individual mice, and you'll be, I hope you'll be convinced with me that these CAR T-Regs can significantly interfere with depigmentation. That also means that we saw more of the TRP1 positive melanocytes being retained in the presence of these T-Regs, and that's being quantified in this particular slide. That's just to prove that it really is containing the vitiligo, and that there are more T-Regs in the skin after adoptive transfer of CAR T-Regs. Not only general T-Regs, but we also quantified the CAR T-Regs, as you can see here on the right-hand side. That figure matches very nicely what the overall T-Regs increase is shown on the left. At the same time, we feel that this is a safe treatment because if we look in the circulation, we don't see any influence on T-Regs, and we are not so worried about general inflammatory responses in the circulation, because CAR T-Regs can be quite aggressive, or CAR T cells in general. And so we did need to check for safety of this treatment. So really what our intent is then that we hope to isolate T-Regs from patients that we introduce our CAR construct into, and this is really on a retroviral basis so that they keep being expressed for a good amount of time. And this is really a therapy that's also, when it comes to effector T cells, is being used for tumor patients as well. And then we want to sort out our T-Regs in a more clean fashion than we've even done here, so that that's all that we're introducing back into the patient, and then follow up on the interference with depigmentation in the patient. So if I can summarize these data, what we tried to show is that patients, vitiligo patients, have fewer T-Regs in the skin, and that also affects the non-lesional skin. So as soon as the T cells, effector T cells come in, there's already the chance for them to do damage. That the same findings were also exhibited in our vitiligo-prone mice, that we see reduced abundance in the skin that can be overcome if we administer rapamycin, but we can also perform adoptive transfer of autologous T-Regs. That replenishing T-Regs can delay depigmentation, and that antigen-specific T-Regs can act on site, and that there's really, we know we have the great advantage that in vitiligo, we know several suitable targets that we can go after that can serve TCR, transgenic T-Regs. We saw that these latter T-Regs to tyrosinase can reduce depigmentation, but maybe not to the extent that we had hoped. But that repeated injection of CAR T-Regs to DD3 does bring T-Regs to the skin and maintain, and that these, in mice at least, we saw that we can maintain pigmentation and melanocytes within the skin. And it's good to keep in mind that with our collaborators, we've shown that you can expand these T-Regs and also store them for future applications. So some of this work has been published fairly recently, and also reported for cutaneous autoimmune conditions where this type of therapy might be of interest. So I certainly haven't been doing this work alone, and I've been collaborating for a long time with Dr. Mehrotra. He's at MUSC in Charleston. And another person who's been very important for this work is, you see him on the right at the bottom, is Jusip Mukatiev. He's now back in Kazakhstan, actually getting married this weekend. And he's done his Ph.D. studies in my lab on this work. So it's been very exciting to work with him. He's gotten a lot of work done. And that's really what I wanted to talk to you about today. We're very grateful for the funding that we've received for this work, and I'm happy to take your questions about this work. And I will leave it at that.
Thank you so much, ma'am. It is indeed a very fascinating experiment and the therapies that you talked about. So, basically in this sequence only, may I ask you a few questions? I have a few questions of my own. So, when you talked about, so in essence, what we have seen here is that you could take out regulatory T cells, you can expand them in the laboratory and to a specific antigen they are directed, and then you can infuse them back into the body. So where are we in terms of human experimentation, ma'am? Has it reached at any level, or is it just being planned as of now?
For now, it's been planned, yes. I mean, we're first going after the IP, so we're going after a patent application for this particular application of this construct. I mean, GD3 CAR T cells have been at least tested, not so much for human use, or at least there's a clinical trial open, but patients are not being recruited in that case at this particular point in time. But that's that progression to the in vivo studies would have to come from, with some funding. So that's what we're looking after besides the IP. But we are very interested in testing this, and certainly, especially in graft versus host disease, the use of antigen-specific T-Regs is really making some headway, because you can, use even T-Regs that recognize allogenic HLA, and that can help to distinguish between, the graft and the host, if you will.
Yeah.
So that's actually the collaboration that we have with Dr. Leventhal is based on that work, and he has the approval and the protocols in place to go forward with these T-Regs for human use, and that we hope to work with him to bring this to a clinical application.
So that's indeed encouraging. Another thing, small doubt that I have, ma'am, is it really possible to use this regulatory T cell graft that we are giving? Is it possible to apply on the surface of the skin rather than infusing into the body through the blood? I understand that they would be transfused or infused in the blood.
Well, yeah, you wouldn't apply them on top of the skin, but it may be possible to have a more localized application, because obviously where we want to stop the response is exactly in the perilesional skin.
Yeah.
I'm not so worried about the number of T-Regs that you would need for adoptive transfer, so I think we can do it in an IV mediated fashion. But certainly, you would need fewer T-Regs in that particular case. And that in itself, of course, also helps, in case you would want to introduce general T-Regs, that you might do that locally. Though, again, in that regard, you would still have to be a little bit concerned about any other responses that you're stopping with more T-Regs present. So,
and finally, one more question, ma'am. Do you foresee any adverse events following such therapies?
Well, I think we did want to make sure that we don't want to interfere with responses that are desirable. But that's exactly why that was the main reason why we want to go after antigen-specific T-Regs. because there was that concern, if you use T cells for adoptive transfer in tumor patients, obviously there's a very different condition to go after where you want to save someone's life. But in this case, you want to improve a life that is hopefully, a long way ahead for us. So you want to be very careful that you do no harm. And for that reason, I think adding antigen specificity adds an extra safety measure to the treatment. But for sure, it's a more comfortable treatment than, there's some things that are coming to market now that could have some serious side effects and where we have to be a little bit maybe more concerned than with a treatment such as this one. So.
Yeah. Sure, and I think, ma'am, if we have a topical formulation, then we can do more work by inducing these, reducing depigmentation locally. So that is something which the patient can do at home. So home-based therapy becomes important in vitiligo patients also. So I think I would invite all our audience to maybe go through with this, and if they have any queries, I understand Dr. Abhishek has a query.
Okay.
Evening, ma'am. Again, this is Dr. Abhishek, and I think permit me to congratulate you. We are informed that you are the proud recipient of the Myron Gordon Award,
Yes.
and additionally, you have been awarded by the American Skin Association recently.
Also, yes.
So
Very excited.
We are kind of privileged to have had this wonderful talk. Permit me to go on a slight tangent, ma'am. What role of the microbiome is there in this entire milieu? If you could enlighten the audience, and do probiotics have a role to play in vitiligo?
I very much believe so. I think you may have seen that I've been very excited about doing microbiome work. We've focused in the beginning on, again, looking at if there's any correlation between the developments in the microbiome. And in this case, we were, kind of a little bit of a step away of some other studies that were being performed, because I think it's kind of a combination of what's happening in the gut and perhaps even, what should I say, behavioral differences, that can exist between patients and controls. That would lead to a different microbiome, can be both the skin or the gut, and to see what correlates with reduced or enhanced depigmentation. And in our case, we administered our antibiotics orally and then followed depigmentation. This is again in mouse models of vitiligo. And then saw that there was quite a difference. And we've also tried this topically. And again, with antibiotics, we could influence the development of vitiligo, in this case in the mice. And we're also following up as we speak on patients and their microbiome, looking at again, environmental factors that could influence this, if you want to, and dietary factors. So I think this is an exciting area of research, and that it could really be very influential. If you're talking about, topical treatments that patients can apply at home, if we can think about, antibiotics, that's such a common thing, but you have to be careful that you pick the right one. So I would still want to make sure that it's, at some point at least followed or, incentivized by dermatologists, because you don't want to apply just any antibiotics. Some actually have the exact opposite effect, because it's really about which microorganisms you're promoting. So Bacteroides seems to be a relatively beneficial set of bacteria, whereas you don't want, things like Pseudomonas, in our case, was what we found in the mouse model. Of course, that's really after a very stark treatment. But I think we are seeing that those are the Bacteroides is something that you'd really want to promote, whether it's in a skin setting or in a gut setting. So we're hoping that we can find, report more on this as we go along.
Right, ma'am. Thank you for the answer. And I think we can take a few more, Dr. Saurabh.
Yeah, we can. I don't see any queries in the chat box. If anybody in the audience wishes to ask anything, they're most welcome. And just to inspire and spread awareness about vitiligo, World Vitiligo Day is celebrated on June 25th and 26th. It is being celebrated for the last 10 years on the day of death of Michael Jackson, who suffered from vitiligo. So this was to commemorate that. So I understand that this is a very basic science research. So yeah.
We hope not. We hope that we can translate this.
No, this is translational actually, ma'am. This is, I would say this is wonderful to know that you can actually transfer T regulatory cells, and there are so many other things that you can do with them. Play around, make them more antigen specific. It's quite intriguing. So I think if there are no more queries now.
Dr. Saurabh, there is, I think, hand raised by Dr. Suman Patra too.
Okay. Yes, Dr. Suman, please go ahead.
Yeah, thank you, ma'am, for the very nice presentation. Ma'am, these days, like normally we are treating the vitiligo with suppression of immunity. So I just want to know whether there is any difference in T-Regs cell expression in vitiligo which is spreading or which are stable. So that sometimes what we see, ma'am, after we stop the drug, again it started spreading. So if we go to that level that we can see that T-Reg cells are still there, are still deficient in these areas, so we can continue the drug for a bit longer so that it doesn't come back after stopping the immunosuppression.
Right. Well, so keep in mind that in this particular case, we're really stepping in at the point where, you know, your effector T cells are trying to kill off melanocytes, and that's when you need those T-regs to stop them. I mean, in other cases, you know, in controls, the T-regs are essentially always there to kind of, you know, to perform immune surveillance in the sense that they want to make sure that any ongoing immune responses are stopped when they're no longer needed. So that deficiency that we see in vitiligo patients, we found whether there's active disease ongoing or not, right? Even in the non-lesional skin, we saw fewer T-regs. So the skin is already less prepared for an effector T cell response. And that's, I think, yes, you could say you would want to have the T-regs there even to prevent further disease. And I know that that's a very important point for patients. But I think we have other approaches that would work in that sense. So this is really where you'd want to step in as soon as you see that there's disease activity. This would be the point of adoptive transfer. I believe that we would want to go after this in a more systemic fashion because that would give you access to all areas where progression is taking place. But theoretically, you could also, if it's very localized, you might be able to inject it on site. You know, if you're talking about prevention, I'd rather step in at a, and I don't want to promote, but that's just one of the ways in which you might think about prevention would be maybe with our modified HSP70. That would be one of the thoughts, but certainly there's, you know, like for example, JAK inhibitors are closer to what we're trying to accomplish here, which is to interfere with effector T cell function. And that's, you know, so it's a different step in the response where we're jumping in here, if that makes sense. Perhaps you could, you know, even though there's not necessarily an overall deficiency in T-regs, but that doesn't necessarily mean that you wouldn't be able to do something about it by enhancing the percentage of T-regs in general. Again, there would be some concern perhaps that you're stopping responses that you do want to occur. So that's always a little bit of a balance, and, you know, weighing, making checks and balances depending on what's most important to the patient at that point.
Thank you, ma'am.
Sure.
I think merely because vitiligo is such a complex disease and there are so many pathways working at inducing vitiligo. Maybe there are autoimmune pathways, then there are redox reactions, then there are genetic factors. So it's a conglomerate really of multiple factors. And what we are looking at here is one of the factors. So maybe if one of the factors is very prominent in the patient, then definitely these are very encouraging times if these therapies work actually in the patients also. So
I saw
yes.
one more question, I
Yes,
think.
yes, Dr. Anil, please.
Yeah, hi, Dr. Caroline. Good morning, and thank you
Hi.
for the fascinating and nice presentation and enlightening as well, actually. So
Sure.
T-Reg is a very exciting phase and definitely in the translational research, basic science research. How soon do you expect that these things will be commercially available as a topical or systemic therapy?
That depends on whether you want to invest a ton of money.
Yeah.
That's really with any therapy. I'm not, you know, I think one of the encouraging things that we know now is that pharmaceutical industries are getting interested in trying to treat vitiligo. Now that sounds a little bit like, you know, why would they not be interested? Because there's definitely enough patients to worry about. But I think for the longest time, there was the concern that, you know, you can halt the disease, but you can't necessarily treat patients to reduce the existing vitiligo, and that you know, industries were concerned about not being able to overcome the already existing lesions. That's one of the things. I think another, you know, I mean, well, that it helps. As I think one thing that you did say that struck me, Dr. Singh, was that you know, there are multiple factors involved. Now, I do think that all patients have an autoimmune response involved in the development of vitiligo, but you also need a genetic predisposition to be there. So it's not like everybody will get vitiligo if you impose the same conditions. It really is built on the fact that you already have that genetic predisposition. So it can always come back in that sense. You can restore the pigmentation, but we know that patients are always sensitive to developing new lesions. And so, you know, there's, I think, now there's an interest in treatment now that we know that we can, even by interfering with this autoimmune response, you leave room for the melanocytes to repigment the skin. Either they do it by themselves, or there's new transplant methodologies, as we just listened to, that can really help with that process. And I think interfering with that autoimmune response will give those new melanocytes a chance. So it's a cool time where industries are interested in this, and I think that will really help get these therapies translated and get them to the clinic. So
Yeah, thanks, Caroline. Thanks a lot. Maybe vitiligo is just a geographic specific problem. Caucasians don't consider vitiligo as a big problem, and India is the biggest problem. So when we have a strong response from the whole world, then definitely we can do something or do more. Thank you, Caroline.
That's why it's very exciting to present in India, because I know that it's taken very serious there, and that's a good thing, because it really helps us understand and treat and develop treatments. So I'm excited.
And actually, it's associated with a lot of psychosocial problems, actually.
Oh, sure.
It's very stigmatizing in India, in all the darker races, I suppose. So that's also one of the dominant reason why we take it so seriously.
Sure.
Part. So,
absolutely.
Yeah.
No,
so, thank you very much for accepting our kind invitation, and we are really honored to have you, and looking forward to work with you in future too. So I understand that our time is up, and we can close this session, and maybe we can continue our communications in future on email and other things. Yes, Dr. Abhishek, shall we close this then?
I think, and it has been an absolute privilege, ma'am. Thank you.
Thank you. All right. Bye.
Thank you, ma'am. Have a nice day.
Thank you.